thank you all for coming to today's talk. I am really looking forward to hearing from Fei Wang. We haven't spoken in a while, but I'm very familiar with his work, and he's been very active in the area of digital health. To give some background, so Dr. Wang is currently a professor at Cornell in the medical school. But I have known of his work even before he went there because he has always been active in the area of medical informatics and AI. You know, he was one of the first to look at EHR data, which is extremely messy and a huge challenge for people who work in machine learning and AI. He has branched out to look at a lot of different medical applications and machine learning techniques to see how they can be used and evaluated in the context of his collaborators at Cornell Medicine. He is a professor in the Division of Health Informatics and is the founding director of the Institute of AI for Digital Health. So now you know a lot about his background. Let's find out more about his work. And you may have heard that we will defer questions to the end, but you can, we'll have time for discussion at the end, and you can also put questions in chat and I'll monitor it there. So Dr. Wang, I'll turn it over to you.
Great. Thank you, Diane, and thank you for the invitation. And because I'm not very familiar with the background of this crowd, so I'm thinking of it might be more productive I give more broad overview rather than the particular topic don't go into very deep. So like you can see what I am currently working on, and more of like from the viewpoint of the breadth. And if there is any particular topic, different people may have interest in different topics, we can always follow up and see how we can maybe build collaborations or follow-ups going forward. So I give as you can see from the title, so I'm a firm believer of going forward, the future of we can call computational health or AI in health would be a more holistic type of pipeline, in a sense that we want to integrate information from different cohorts and different modalities rather than just focusing on one particular aspect. So certainly that raises a lot of challenges, and you know, there are a lot of you know, national and international efforts tries to achieve this. I think two well-known efforts would be the UK Biobank and also all of us. But we're going to see more and more, and I think that will be the future, and also there will be a lot of methodological challenges for us to innovate as well. So something about myself, my background, as Diane introduced a bit, I am trained as a machine learning scientist and then did a postdoc in statistics. I started working on health since 2010 when I joined IBM research, and then in 2016 I joined Cornell until now, and last year I founded this Weill Cornell Medicine Institute of AI for Digital Health. So when I say I don't think I need to introduce too much about AI and machine learning. I believe you guys all know that. But when I say digital health, it's not just what we usually talk about, because when people usually talk about digital health, the things came to mind as wearables, mobiles. But what I mean by digital health is really anything that involves digital components and you know, capture data, store data, analyze data to achieve more personalized and precise medicine. And certainly it's going to involve a lot of techniques and also a lot of different types of data. And I like this figure a lot. So in order to you know, precisely recognize the condition of a particular patient or a particular person at a particular time point. So we really need to understand from all different angles. It's not just the genome. In the past, when we talk about personalized medicine, people really means on the molecular level, the genetics profiles of the people are similar or unique. But later on turns out it is not enough. We also need to look at the phenotypic manifestations, whatever reflected in the clinical data and patient reported data, even patient survey data. And also exposomes becomes more and more important with, I think now there's a hot topic on study what is the influence of the climate change, especially the heat, gonna impact the people's health conditions like cardiovascular, cognition, all these different kinds of things. So it's the health of a particular person is really complex composite, reflective of all the changes from all those different aspects, from really molecular microscopic level to all the way up to the really macroscopic, the exposome level. So you will see some of the work we have done, try to achieve this, but I would say we are still pretty early on the road. But I believe, as I said at the beginning, this is a path forward. So I will made my presentation in the context of COVID through a series of studies. I know one of the main interests from this group is cognition and neuropsychiatry related stuff, but I hope from the examples of COVID, I will make some analogies. So we also have done a lot of parallel studies in other diseases, including Alzheimer's and Parkinson's. So I hope I can draw some parallels. So the same problem or same technique or same setup also applies to those diseases as well. I will also have a slide summarizing some of the ongoing projects in particular on Alzheimer's disease and Alzheimer's disease related dementia. So the reason why I introduced COVID is because we all know, like, since the pandemic starts in New York City is first is epicenter at the very beginning in the first wave, and the doctors and clinicians and whoever professionals suffer a lot of pressure at that time. So I am in a medical school, and we have our hospital, and at that time we have a lot of demands on, really in a short period of time. If there is any technologies, we all do AI, do machine learning, but is there anything we can do to help to cope with or face with this challenge and come up with better solutions? So there is a real need. We have done a lot since 2019, even until now. We're still writing papers just this week to study the effectiveness of Paxlovid for long COVID. But it is ongoing because this is a disease that is still not known too much to people. So that is the reason why I introduce we do research on this disease, and I want to introduce in this context. So at the very beginning, at New York City, because at that time the number one, the resources, pretty limited resources, not just in let's say the number of beds, the number of clinicians, but also, or the number of equipments, but also you all know, like nowadays we confirm COVID-19 or SARS-CoV-2 infection by a particular lab test called the RT-PCR test. And in order to do the PCR test, you need a particular chemical. And at the beginning, the supply of this chemical is not that rich. So which means that it is not like today, everybody, if you want to take the test, you can take the test. At that time, the policy is you have to demonstrate certain relevant symptoms like fever, like cough, and then you can order the test. So what is the consequence? So an immediate consequence is, number one, the positive rate is very high. So in March and April 2020, the positive rate is 50%, 50. And certainly, like there are a lot of demands, and plus during the first wave, the acute outcome is really bad. Lots of people ends up in the hospital in the ICU, got intubation and prolonged intubation and death. So our colleagues at laboratory medicine who are in charge of all those blood testing approached us. I mean, with some requests based on some of their observations. So first, talking about the challenge. Number one, the turnaround time for PCR test at that time was 24 to 48 hours. So you take the test, about 2 days, you can know the results. Number two is, I mean, now it's still a challenge. That is the PCR tests that have a relatively high false negative rate. So false negative means that the result is negative. You let the patient go, but actually the patient is positive. So that's gonna make the pandemic more difficult to control. And the third thing is because of the reason I just mentioned, so people demonstrate the relevant symptoms, so they can get the PCR test, which means that at the time when they get the test, they already probably in the course of, if there is an infection, already in the course of the disease development. So what happened is those people are typically not just took the test, but also took a bunch of routine blood tests. And our lab medicine people discovered that for people who actually got SARS-CoV-2 infected later on, they typically going to demonstrate some abnormalities of those routine blood tests. So they say, can you machine learning people help us to develop a model that based on routine blood tests to predict what could be the infection outcome. So there are multiple benefits if we can do that. So number one is routine blood test is much widely available because it is routine, it is cheap. Every hospital has that. It is not like PCR tests. Number two is the turnaround time of routine blood tests are pretty fast. So you don't need to wait for 2 days to get the results back. And number three is hopefully with these indicators in the routine blood test, it can also catch some of the potential false negatives. So with very good motivations, and we collected the data from, we are affiliated with New York Presbyterian Hospital at Weill Cornell. And we collected the patient data from our hospital for training. And we also use an independent set, which is the New York Presbyterian Hospital at Lower Manhattan, a different site for testing. So the whole procedure is like this. So here is the PCR test ordering time, and we look at 2 days, within 2 days before the PCR test ordering time, if there is any routine blood tests that have been taken, and you form a patient representation vector based on those routine blood test results. So every dimension is a particular blood test. And you may ask, so what if this person have multiple, let's say, white blood cell count? So we just do some summary statistic, like you can do average, you can do mean, you can do max. We did some sensitivity analysis, doesn't seem to like impact the result too much. So essentially, we use a routine blood test, results as the descriptors of the patient in form of vector, and then we use that to regress or to classify or to predict the binary outcome if the patient's finally got a PCR positive or no. And we tested a bunch of models, including simple models like linear, like logistic regression, and the simple decision tree, and the relatively more complex models like a random forest and gradient boosting tree. You may ask, why not use deep learning? I mean, first, the sample size is not that large. Second, the data is not that complex. We know deep learning, the strength or the power really comes from when you have large amounts of data, very high dimensionality, and the complex interactions may hide in this complex, large and high dimensional data. So you can use deep learning potentially to tease that out. But here the representation, the problem is pretty clear, and there's no such complex and hidden patterns there. So you know, we honestly we also implemented the multi-layer perceptron. We just don't observe. They improve these results. So we just didn't list them here. So that's actually one of the pretty common observation in a lot of the machine learning for healthcare, especially clinical data analysis scenarios. A lot of the times, deep learning model cannot, if you set the setup appropriately, cannot or very difficult to beat simple. A lot of times, even for regularized logistic regression, it is very hard for a complex deep learning model to beat. So one analogy I'm to draw here is this is a very common type of setup when you want to do risk prediction. So you can think of this as also like predicting the infection risk. So I mean, on the Alzheimer's side, people have also done a lot of works like using electronic health records. Here, we only use lab tests, but in electronic health records, you also have like the historical, like maybe it's not 2 days, but in the past 5 years, you predict like what is the likelihood of the onset of Alzheimer's in the next, let's say 6 months or 1 year. So in the historical data collection, you're gonna have not just the lab tests, but also like procedures, medications, diagnosis, or whatever, and just pour whatever in your prediction model and see how well you can do the prediction. And there are some recent publications from different groups like Florida, like Northwestern, I mean, building these models and reported some promising results, but there are lots of challenges. You all know, you do cognitive, you know, disease analysis, like the information may not be precise in the EHR, and lots of the important information may not be captured. All these challenges. So it is yet to be seen if those models are really reliable and trustable. But anyway, I just want to draw that analogy that this is a very common risk prediction type of setup in using clinical data. And as you can see, so here if you look at the gradient boosting tree and random forest, actually the performance are pretty good, which means that it is possible that we can use a model to you know, do such a prediction. And remember, we also have a motivation is whether this model can help us pick some of the false negatives. And this is one example. So this is a person in the EHR records. So this person was tested negative. But within 2 days, this person came back to the hospital and the test turns positive. So which means that the first negative could very likely to be a false negative. And actually we did analysis with false negatives defined in this way, which means that the test is negative and person returned to the hospital within 2 days and changed the result. So we just you know, define those cases as false negatives, and we like for this case, our model predicted this person is having a very high probability to be a positive. And if you know this plot, this is generated from SHAP. It can tell you what is the reason, like the high LDH, that's the inflammation marker. So that is a red flag. So ferritin, that's and also high level indicates there is some inflammation going on. And older age is a risk factor, lymphocyte, CRP, basophil. I mean, all those are suggesting there might be some infection going on. So that's why our model produced such a high probability. So it can give you such a kind of like interpretation. And we scan through all these potential false negative cases, and um, uh, our model can capture 30% of those. So which means that our model predicted it should actually be a positive. So after we developed the model, actually we were almost implement that in our emergency medicine department. But if you remember, so the first wave came down since May of 2020. So, and also at May 2020, the resources are becoming much more available, especially in terms of testing. And actually by the end of May, it's pretty much like today. So everybody who wants to get the test, who can, I mean, you can get the test. So actually the positive rate drops from 50% to 2%. So the hospital, did not have that large pressure, and given also the acute infection outcome is getting much better. So we did not really implement that, but we at that time we already study Epic and find out how to plug this in through their APIs to come up with a kind of like an alerting system to alert the physicians. And our work was reported by a lot of media at that time. So this is just one example. So this is a very typical setup for risk prediction. It's not just in this context, as I said, in a lot of the other diseases, this is one of the core problem for, if you talk about machine learning for health, lots of people just think about predictive modeling, supervised learning, and a lot of time what they mean is really just for prediction, risk prediction. And the second a typical problem we have we frequently encounter in machine learning for healthcare data analysis is the previous one is prediction, but sometimes prediction is not enough. You need to draw some causality relationship. So the previous one is just prediction is association. There's no suggestion of causality. But sometimes, let's say if you want to using real world data to justify or to investigate the effectiveness of a certain treatment. So in this case, you just do prediction that may not work because there could be a lot of confounders. I mean, if a certain person take a drug and getting better, it is very hard to tease out it is the effect of the drug or it is because of other factors. So we typically need to like follow a protocol that's similar to conducting a rigorous randomized clinical trial, where in the trial you can do randomization, but in real world data you cannot do randomization, but you want to mimic that. That's called pseudo randomization. So you have to do appropriate weighting, typically through the inverse propensity score. The propensity score is the likelihood of the patient will receive the treatment, and then use that way to balance the covariance and also balance the and also weigh the outcomes to have a more objective assessment on the effectiveness of a treatment. So this process has a term that is called target trial emulation or trial simulation. So we actually, this is one example. It is not investigate a treatment, but investigate. So people talk a lot about long COVID, but there is until today, there is still no standard definition or diagnostic criteria or on what is long COVID. So we want to define, we have a national cohort and we want to define, so what is long COVID? So essentially we look at for patient who got COVID and after the acute period, which is typically 30 days, within the onset and 30 to 180 days at the post-acute period, we look at if there is any new condition appear in that period. And we compare the COVID positive patients with the COVID negative patients. So here the exposure is not really treatment, but the COVID infection, and the outcome is the incidence of a certain condition in the post-acute period. So we still follow the target trial emulation framework, and we do that in a high throughput way. So we investigate hundreds of conditions and look at like if certain conditions have a much higher hazard in COVID positive population compared to the COVID negative population. And you can see, we observe in a lot of, a lot of conditions covering, quite a diverse set of organ systems. We through this framework, we want to draw, I cannot call it causal, but at least a pseudo-causal type of analysis to really see, these conditions could be due to the exposure of COVID. But certainly there are lots of challenges for EHR, like missing information, noise and all of those other things. We cannot rule out all the potential residual confoundings and all these things. So, but in this case, what I'm trying to say is we need a different framework other than the prediction to achieve the goal of objective assessment. Actually, we have another paper using exactly the same pipeline to investigate if there is certain drugs that could be repurposed to Alzheimer's disease and Alzheimer's disease related dementia, also published on Nature Communication. So this same type of pipeline. The goal is to draw some pseudo causal relationships can be applied in different scenarios. So that's the second type of very typical problem we encountered in biomedicine. And the third one is clustering. So a lot of the diseases, including Alzheimer's and COVID, and in this case, what I'm showing here is long COVID. So they are heterogeneous. Heterogeneous means that for people with the same disease, they're disease pathophysiology and of course the clinical manifestation could be very, very different. So that's also one of the reasons why people talk a lot about personalized medicine or precision medicine. But that's extreme in the future. Like we really want to tailor the treatment for particular patient at particular time. You need a lot of data to achieve that in a robust way. But in between, so one of the intermediate step is can we identify stratification or strata of the patients such that the patient within the same group, they have some common disease manifestations. Let's say in this case, we just look at the clinical data. So that's why we call them the subphenotypes. So the patient within the same group, they have a similar manifestations, like similar disease and medications. You will see later. These similar phenotypic demonstration may indicate some common disease pathways, and that may be suggestive of a particular type of treatment or a management way. So that can inform, of course, the disease mechanism study, deriving appropriate hypothesis, and also inform public health policymakers to make more stratified or targeted policies to manage different type of patients. Let's say in this case would be long COVID instead of one size fits all. So in this case, we still, follow the setup of the previous study I showed you. So we look at the new incidence of conditions for COVID positive patients in their post-acute period. So you can think of, let's say I have 200 conditions. So then I look at if any condition is a new incident in the patient particular post-acute period. If a particular condition appears that is then I have 1 on that particular dimension, and if not, then it's 0. So every patient is represented by a 200-dimensional binary vector with 0 and 1, indicating if that condition is incident in the post-acute period. And then I do clustering. So based on these 0-1 patterns, so hopefully after the grouping on top of this, so the patient within the same group, they have similar patterns of these 0 and 1, so which means that the patient tends to have a similar incidental conditions. But the reality is the data is really messy. So, and you cannot, that's the first step we try. So it is almost impossible to tease out very clean clusters just based on these high-dimensional binary vectors. So what we do is we draw, again, we draw an analogy. So we thought, so this is very similar to the situation when we do text analysis. So that's there is one topic in machine learning called topic modeling. So really, you represent text pieces with words, and you know, these text pieces from different topics. The assumption is they have different representative words. So if we can identify the topics, that's essentially as the groups of text pieces that sharing a similar word appearance pattern. So that's why we call it the topic. And similarly, I mean, when you use words to compose a text piece, so there are lots of challenges. Some word are very common, some word appears here and there in a more random way, and there are some keywords. So we have exactly the situation, the same situation here. So we use topic modeling to learn the hidden topics and use the projection onto this topic space as the new representation of the patients, and then do clustering on top of that. And then we identify 4 pretty robust subphenotypes of long COVID. So one covers cardiac and renal problems, and certainly these problems a lot of times are more severe compared to the others, and we find a lot of the people from within this subphenotype, they got infected in the first wave. And in the first wave, you know, the patient typically suffer from more severe acute outcomes, and there is a hypothesis for long COVID that is, you know, hyperinflammation. So it could be, you know, the very bad inflammation happened during the acute phase that causes some damage of the organs, and that leads to more severe long COVID manifestations. And subphenotype 2 is a pretty normal. It's a really respiratory problems and sleep. And for 3 is musculoskeletal and neurological systems, like lots of people suffer from headache, neurological pain, and for doctors in the clinic, they see a lot of patients complain about these. And something interesting is we find a lot of these patients at the baseline, they have some autoimmune problems, like they have asthma and rheumatoid arthritis, this kind of thing. I mean, nobody knows what's the linkage there. And subphenotype 4 is digestive, more of digestive. Respiratory is pretty common. Digestive means that people have constipation, have diarrhea, and there are recent you know, mechanism studies suggesting that the SARS-CoV-2 virus can stay in your gut for as long as 7 months, even you know, after your the virus is cleaned in your lung. I mean, they can still exist in the gut. So that's another type of hypothesis called the virus reservoir. So that could be explained, but we clearly tease this pattern out, and we actually use a large New York City-based cohort for development and use Florida-based cohort for validation, and it validates pretty well. So these actually teased out the major patterns of long COVID, and as I said, it can trigger follow-up mechanism studies for basic science folks, and also can trigger some public health policies. And the same thing, actually, we also have other works on trying to subphenotype or subtype Alzheimer's disease and Alzheimer's disease-related dementia, because that is also highly heterogeneous, and hopefully that can tease out some potential subtypes that trigger a more stratified treatment. And certainly, I mean, whatever I have introduced before are all based on the clinical data, but more and more, that's the type of data I have most of the experience. But with the research going on and on, I frequently got asked, like, for example, for the previous question, like, you discovered those 4 robust subphenotypes, which is very good, but then so what? How can you validate those 4 subphenotypes really make sense? Here, validate people really expect to see if there is any clues from the molecular angle. If you can find, let's say, different genes or pathophysiologies for different subphenotype, that's perfect. So then, I mean, I'm thinking of it's just using the clinical data may not be enough. So that's why, I mean, over the years, we have also been trying to analyze molecular data. This is just one example of analyzing multi-omics data, single cell multi-omics data from COVID patients, and we developed again, this is a hybrid topic model we call multi-omics embedded topic modeling, to hopefully integrate both single-cell RNA-seq and single-cell ATAC-seq and the proteomics to hopefully derive a joint embedding from multiple modalities of the cells, and then hopefully that can reflect some intrinsic properties of the cell that, as you can see on the right panel, with some simple clustering, it can reflect the ontology of the cells in terms of the types, as you can see. So this figure is a more broader scale, that you can see the different cell types are clustered together. I mean, this is from COVID patients, and this is within this B cell. So you can see the different subcell types can also be teased out. And we did some differential gene expression analysis and find out a particular gene, IL-6, was consistently expressed at high level among B cells, including the malignant subtype, so which suggests a mechanism, potential mechanism why cancer patients that typically got more severe after COVID infection, typically got more severe outcome. And this can suggest, IL-6 could be potentially a gene target. So this is just one example, certainly. So analyzing those data needs, our basic science lab friends to, collect the data and, because these cannot be like in clinical data. These comes with a professionalism and also a cost and also time. So a lot of times these data in terms of sample size is small, but of course in terms of number of cells is large, but in terms of the patients is typically very small. So, but this is something we are also touching, and because this is important in terms of understanding disease mechanism, and we actually wrote a perspective in Lancet Digital Health during COVID, and talking about how to integrate the data from different levels, hopefully to derive hypothesis on the drugs that could be potentially purposed to COVID. If you remember, a lot of the treatment like remdesivir is developed. Original indication is Ebola, and the baricitinib, which is discovered from benevolent AI by their knowledge graph, is a drug. Original indication is autoimmune. And dexamethasone is, for the treatment of severely infected patients in ICU. All these drugs already existing, and people explore their utility in treating COVID patients because that saves time compared to a brand new drug designed on molecular level all the way past clinical trials. It's gonna take a long time. I mean, we just cannot wait for a pandemic like this. And last but not the least, we also have some studies like the left paper is one of the very first study on like the impact of temperature and the humidity on the transmissions of SARS-CoV-2. Because at the beginning, if you remember, people were talking about, oh, I mean, it is winter when it comes to summer. The weather, the temperature is increasing and the humidity is increasing, and maybe the pandemic will end automatically, and we collect a humongous amount of data, public information from the internet, from United States and China. And we show that just purely based on, temperature and humidity, it is not enough to reduce R naught value to something below 1. So that is the necessary condition for stopping the virus transmission, but we just show it is not enough. So I mean, just to depend on the climate change won't be enough to stop the pandemic. And on the right is we show that for long COVID at different geographical area where you have different environmental risk of exposure to certain chemicals or climate conditions, that people will have differential phenotypic demonstrations of long COVID. So these are kind of like for people's condition, you also need to consider the impact of the environment. And as I said, so those are examples of COVID, but we also have a series of lines of research on Alzheimer's disease and Alzheimer's disease-related dementia, and on the mechanism. So we are working together. We have a Appel Alzheimer's Institute with basic science people, and they have some hypothesis on this particular genetic variant, and we have a joint project. Hopefully can test the validity, and also my role will be to show using real world clinical data that we can derive some potential treatment targeted this genetic variant and show its effectiveness. This project is still ongoing at electronic health record. So we know for most of the Alzheimer's disease trials, they failed. One of the, I mean, there are lots of potential reasons for that, and we hope to explore the electronic health record data to not just to derive hypothesis, but also help refine the design of the clinical trial. So we have 2 R01s on this topic, and this is Nature Communication paper I mentioned, using causal inference and real-world data to derive hypothesis, a repurposing hypothesis for ADRD. And certainly the impact of environmental and social factors is also important. So we have 2 R01s on this. So one is studying the role of environmental factors and social factors on impacting the progression pathway of Alzheimer's disease, but in a particular population, which is sexual and gender minorities. And another R01 is to study the impact of environmental factors for patients who got COVID and got cognition-related impairment as long COVID, and what is the role of environmental factors in impacting that. And certainly another thing is, like diagnosis and treatment and whatever for cognition related diseases is pretty challenging in terms of like neuroimage and get protein markers. Those are pretty invasive. So anyway, we can use low cost and more accessible technologies, that would be always appreciated. So we have another R01. This is a paper long time ago, long before ChatGPT. So we developed some chatbot using simple reinforcement learning, using the data collected from Oregon Health and Science University. They have large trials collecting the conversation script between the patients and the clinician, and the hope is to use an agent to replace that conversation, and so that the agent can, through a conversation or dialogue with the patient, can reach some conclusion with the least number of rounds. This patient is more likely to have some cognitive problem or no. So that can, if we have, can serve as a screening tool, potential screening tool. And that is also leads to another ongoing project. So as you can see, so we kind of like also try to touch all those different aspects. And actually, this figure shows a more holistic picture of the ongoing efforts in terms of the different types of data that in my institute. So as you can see, so we really try to integrate the information from all different places. And certainly the challenge will be, how are you going to integrate that? And this knowledge graph in the middle really serves as that role, where you have all biomedical related entities and their relationships, and this is curated from existing knowledge, literature, guidelines, that can hopefully link up the entities that you can see or encounter in different types of data, and potentially associate them. And we actually have built this integrative biomedical knowledge hub, and we made this publicly available. You go to the GitHub, and all the source files are there. And actually, not only that, we also build kind of like a user interface. I have actually a video here. Let me see the time. I think I still have time to play with this.
Biomedicine has an enormous volume of highly specialized knowledge accumulated from various sources. Here we present the IBKH, a knowledge base which has harmonized data from 18 resources. Let's say you're interested in finding novel treatments for Alzheimer's disease. You can begin by generating a first neighborhood subgraph of Alzheimer's using the Entity Query Search tab. Note that there are multiple entries for Alzheimer's which are indicative of different types, which you can learn more about by hovering over the information icon. Right now, we're looking at the top 200 connections, but this can be changed via the limit. To find drugs that currently treat Alzheimer's, let's go to the Path Query tab. We input Alzheimer's and a relation of treating drug and add it to our path pool. You can add more than one path to this pool simultaneously. You can then visualize the drugs that have direct connections to Alzheimer's. At the top, you can find the cipher query that lets you retrieve data from the graph database. You now want to find novel drugs that could treat Alzheimer's. Our knowledge discovery module uses graph embeddings to predict the most relevant entities to the input. Let's input Alzheimer's and submit. Looking at the drug section, we can see that sodium bicarbonate is a link predicted by the model. By looking at the evidence, we can see the path between the drug and Alzheimer's. Finally, you want to explore some characteristics of a cohort of Alzheimer's patients who have TREM2 mutations. The IBKH allows users to input multiple entities describing their cohort. Our model will predict knowledge in the IBKH that is associated with the input cohort and the evidence for those associations. All in all, the IBKH provides innumerable ways for users to investigate and generate evidence-based hypotheses.
All right, so this is an introduction of our software, and actually the user interface is also made publicly available. We put the instance on AWS. You feel free to play with it, but this is continuous maintaining efforts where currently I think it includes 18. We consolidate 18 different biomedical knowledge graphs, and there are a large amount of work in terms of consolidation. We need to normalize all the entities and the edges and all this kind of thing. But we continuously merging in more knowledge bases, knowledge graphs, and we also hope to add in new type of entities, like from medical images and so on and so forth. So it will become richer and richer. But that is a pretty important resource for us to, especially in the role of linking up the heterogeneous entities from different domains. But whatever I have introduced so far are more on the application perspective and data perspective. Certainly, I mean, as you can see, it's a super complicated, a high dimensional, sparsity, temporal, heterogeneity, whatever. So it needs innovations on the machine learning side. So some of the important aspects you also see lots of the times in recent years when people talk about machine learning in health, especially on the technical side, these are kind of like some popular topics people talk about. I mean, I just put in some of my papers there. So this paper I wrote with my colleagues. It's kind of like 4 or 5 years ago. We envisioned like the challenge and promise of applying deep learning in medicine in a major medical journal, and privacy. So people talk about how to incorporate more data, but without leaking the privacy. That's federated learning. We have this perspective piece on federated learning, its potential in healthcare. And people talk a lot about fairness. Fairness is not new concept, but what is new for AI or machine learning model in fairness. So we also have this review piece explanation. So people by default expect the model should be explainable in medical setting, but that's actually not the answer. If you consult medical doctors, a lot of them says, if you can show me robust evidence that the model can perform uniformly well in different scenarios, I'm comfortable using that. And one of the example they use typically is on drugs. A lot of the drugs, we don't know the mechanism of action, but we still prescribe that, like aspirin. So I mean, so do we really need interpretable or explainable machine learning model in medicine, or black box is fine? So I wrote this piece to talk about what we really need is like a level set the risks. So in different scenarios, we may need different requirements on the explainability or interpretability of these models. And the variety is really, you already saw, like from those different information sources. And there are also lots of papers. I mean, we as well combine, because all of these aspects are important. So it is hard to say, so which one is more important than the other. So, but we really need to think of how to develop a model that can, you know, satisfy all those requirements. And we, for example, this is in Europe's paper. 2 years ago, we look at a federated learning mechanism guaranteeing not just the performance, but also the fairness. And this is a KDD paper, I don't know, of 3-4 years ago. We tried to explain the algorithmic fairness or disparity through what we called fairness-aware causal path decomposition. So you know, like there is a disparity, what causes, what decision path leads to that disparity, and you can make appropriate adjustment. And the paper I introduced in the topic modeling multi-omics part, we try to build model to, you know, integrate multiple modality. The reason why we choose topic model is because it is interpretable. And we have a recent perspective piece, which I really believe that should be the future. Is it's people talk a lot about multi-model. So the reality is it is not just a multi-model, but also cross cohort. So in reality, we typically have the situation like you have, I mean, for the same disease, let's say Alzheimer's, you have one cohort, let's say ADNI, you have very rich neural image and genetics information. You have another cohort, let's say ADSP, you have full sequencing data, but no neural image information. You have a third cohort, let's say whatever electronic health record data. I mean, very rich clinical information, but no imaging and the genetics at all. So, I mean, all of these cohorts, you know, they have different focus and about the same disease. is there any way we can leverage all of them? I don't think too much work have considered this. People typically only consider for the same cohort with different modalities, how do you do the integration, or with different cohorts but share the same modality, how do you do federated learning? But the reality is like what I just said. So we call this cross-cohort, cross-category learning, or C4 learning, and we propose a strategy called patchwork learning, and we have the iClear paper accepted as Spotlight as the initial step, theoretically, because you need to handle the alignment of different modalities and different cohorts at the same time. It is pretty challenging, not a simple question. That's all the contents I want to talk about. And last but not the least, the acknowledgement. We are funded by a lot of different funding sources, which made our research possible. And this is my current institute, the people. I mean, of course, without their help, I can not do all these fascinating research, and also thanks to my collaborators. And now I think we have 5 minutes for questions.
Thank you very much. That was a lot of different studies you have going on to absorb. And I know there was one person who asked me to pose a question. He had to leave about 5 minutes early.
Sure.
And his question was to ask you to elaborate about your views on personalized medicine and the role of AI along the lines you alluded to in the beginning of your talk. If you could elaborate a bit more on that.
Yeah. As I said, so as you can see, so all of my vision about why do we need to, touch all those different kinds of data is really towards the direction of personalized or precision medicine. You really need to gather all kinds of information from different sources to understand the condition, personal condition at a particular timestamp precisely. But now it is just not possible to achieve that, and because of the limitation, not really on technology, but really on data. So there are some good efforts. I mean, the important one is all of us. And I mean, I cannot tell like when they achieve their goal, like enroll 1 million people, is that enough? I bet it wouldn't be enough as well, but at least there will be a good ground for us to further investigate this. But for now, as I said, like a stratified medicine as an intermediate step is much more achievable. I think in particular in the context of Alzheimer's, one of the reason that people believe why so many trials fail is because the disease is too heterogeneous. So when you recruit patients at the time of randomization, although those people all looked similar, but over time, so they can progress in very heterogeneous ways. That's so-called like people have, I mean, they're a rapid progressor, they are a slow progressor, and those rapid progressor can, a lot of times just destroy the trial. So if there is any way that we can tease out those patterns and more importantly, predict, beforehand, that can definitely help with the development of personalized medicine and also treatment.
Thank you. That's good. I am looking at the participants to see. You can unmute yourself and ask a question or raise your hand. Well, I have one more, and that is, I like your discussion of phenotyping different conditions or people, and you have done that with several different types of data. You've looked at EHR data, you looked at some of the cell data. I'm just wondering how you handle this and topic modeling as an example with a heterogeneity of data, numeric, time series, text. Have you considered that, and what have you found to be successful?
So very good question. So we haven't reached the state of incorporating really heterogeneous in a sense, like both genomic and the clinical data. So like in this case, it's just a single cell multi-omics. So like the different modalities would be a single cell RNA-seq, single cell ATAC-seq, and proteomics. So although it is still heterogeneous, but all on the cell level. And for this, we just look at the clinical data, although we look at both diagnosis and medications, but all within the EHR. So we haven't do crosswalk yet. So there are some ongoing work that we try to tie up really information from, let's say, clinical and omics. A natural thought would be to leverage some graph-based method, but we haven't have anything published or released yet. But that's kind of like the future, because you do need those different aspects to characterize the disease.
Right.
Yeah, I want to say thank you for that presentation. It was amazing as you were talking about your research that's ongoing. I just wanted to know all of the answers now. So I'm going to be looking forward to that. As you were talking, I was thinking, we have a number of students in the audience, and this is so much data you're pulling together. And so I appreciated you showing the hub, but I was wondering if you have like any other suggestions for students, how they might get started, or what might be kind of the types of data sets that, because EHR is very complicated, like, what sort of recommendations might you have for them in getting started with research like this?
Yeah, sure. So first, I mean, for students getting started for research, you have to have a particular PI. You have to join a lab. So a different type of PI, let's say basic science, I mean, not necessarily basic science, but bioinformatics people and clinical informatics people, they're going to have access to different types of data. So, I mean, the preparation would be different. So I think getting to know like what's your interest. I mean, I don't know for WSU, but for Cornell, so like a lot of my students, I think doesn't matter in which PhD program, they're gonna do rotations. So like typically each rotation is 2 to 3 months, depends on the program. So you can join different labs to understand the flavor and the type of problems they're focusing on, and whatever the resources they have. And after 2 or 3 rotations, you can decide so which lab you want to join and work with the professor as your PhD or master as well. So I think getting to know the PI's focus, and also getting to know the data and the resources different labs have is definitely the first step. And then after you identify an appropriate PI, so you can get familiar with the data. Like for me, I have access to a lot of the EHR data, which may not be the case for a lot of other PIs. So if you're interested, if students are interested in working with clinical data, they approach me, I can easily add them to the IRB, and they can begin to explore the EHR data. And you said the complexity is very much true, but I have a lot of junior faculties and also postdocs that are very well experienced with the EHR data, and we have the pipelines that can hand over to the students so they can study, but not really from scratch. I think that can help smooth, especially the start, a lot. And then we also work very closely with almost for every project we have clinical partners. So with domain experts, so we understand like how those real domain experts are thinking of what is a pinpoint of all those problems, and which kind of data you should think of, and which kind of model. I think we not just the domain expert, but also we have like computer science people as potential advisors. So I mean, with all of these support, so students can have a more supportive environment to getting started and get the exploration done.
Well, thank you very much. I know we have trickled over a little bit past the hour, so we'll wrap it up, and thank you for your time, sharing your ideas and summary of your work, and we look forward to digging into your papers and learning more.
Sounds great. Thank you.
All right, thank you.
Bye-bye.
Thank you.